Ladies and gentlemen, Joan Steitz.
Thank you, Rick, for those over-the-top comments. And I want to say thank you to Leah and to the other trustees of the Hope Funds and all of you who are here tonight. I have revered Jim Watson ever since I first met him in 1961, not only as a scientist, but as a very caring mentor and friend. I count myself privileged to have been part of the 20th century revolution in biology. I was a junior in high school in 1953 when the double-stranded structure of DNA was first published by Watson and Crick. And by the time I reached college, it was still too new to have found its way into textbooks or even into courses. Fortunately, as an undergraduate at Antioch College, I was assigned to a work-study job in one of the very few new labs in the field of molecular biology in Alex Rich's lab at MIT. And I recall being completely enthralled when I got there and learned about double-stranded DNA, and this was because it provided a molecular explanation for so many of the genetic phenomena that had fascinated me as a high school student. My project there was on ribosomes, supervised by John Warner, whom we've already heard mentioned tonight. It was my first introduction to RNA, which, as you've heard, would become my lifelong passion in science. Yet, I decided to go to medical school because I had simply never encountered a woman science professor or lab head. Women were research associates in someone else's lab, obviously a man's. But on the other hand, I did know several women physicians. My goals changed only during the summer before I was to enter Harvard Medical School when I worked in the lab of cell biologist Joe Gall, who was then at the University of Minnesota, later at Yale and the Carnegie. By August 1st, I found that I was so exhilarated that I decided that my future prospects did not matter. I wanted to make scientific discoveries and switched instead to the PhD program at Harvard, actually with some help from Jim Watson, whom I had met when I was at MIT. So the new field of molecular biology that I then entered when I became a graduate student in Jim Watson's very, very active and exciting lab was small and eclectic. Everyone in the field of molecular biology knew each other, even though their labs were located in different parts of the world, and they'd been trained in discrete disciplines, such as physics, chemistry, and virology. Molecular biologists were united by wanting to uncover the basis of life in molecular terms. Their focus was exclusively on simple systems like bacteria and the viruses that infect them. It was reasoned that you could never learn anything about the underlying principles without starting simple. And Jim repeatedly reminded us not to waste our time working on something that was not important. Investigators who at that early time were endeavoring to work with higher organisms were even derided, and that's true, for focusing on systems that were too complex ever to be understood. So it was therefore unimaginable to me what would happen in biology within my lifetime. For instance, it was inconceivable that we would ever know the sequence of the 4 billion base pairs of the human genome. A co-graduate student with me in the Watson lab wrote his entire thesis on the determination of the sequence of one nucleotide, namely what was at the end of one end of the RNA genome of a bacterial virus. Had to start somewhere, so you start with one nucleotide. Also unimaginable were the far-reaching implications that the revolution in biology would have for the practice of medicine with new drugs and new diagnostics, and we've already heard a lot about that, and I'll come back to this a little bit later. I could not see that the biological revolution would spawn a multi-billion-dollar, now it's trillion-dollar, biotech industry that would make products useful not only for medicine but also for agriculture, or that it would revolutionize the field of evolution and even of forensic science. Finally, it was inconceivable to me that women would hold important leadership roles in science and academia, which we're now finally seeing. Another surprise was the realization of how important serendipity is to discovery in science. For instance, my postdoctoral project that was mentioned was serendipitously chosen and resulted in new insights into how bacterial ribosomes know where to start on a messenger RNA in order to read the genetic code and make a protein. A decade later, at Yale, a casual remark by another scientist about clinical medicine led my MD PhD student Michael Lerner to use autoantibodies from patient sera to discover a new class of tiny particles in cells called SNRPs. SNRPs are essential for pruning out the nonsense segments that interrupt almost every human gene and its transcript through a process called splicing. SNRPs therefore allow the information in our genes to be converted into working proteins in our cells. Knowledge about splicing has become increasingly important in all the things we've been hearing about tonight, since it is now estimated that more than 60% of human genetic diseases originate from defects in the splicing process, including many, many cancers. Understanding has given rise to novel RNA therapies for a variety of diseases, with the lodestone being the mRNA vaccine for COVID. Thank you so much, Melissa. During all this time, working on basic science, trying to figure out about RNA, I never doubted my conviction that this basic research would contribute to human health. But I also never anticipated that I would live to see research on RNA produce basically a magic bullet for combating a worldwide pandemic. It's absolutely amazing. A final surprise has been that it is almost as much fun to share the joy of discovery with a younger colleague as doing it oneself. I've been immensely fortunate to work with a cadre of very talented younger people, PhD students, postdocs, and undergraduates, two of whom are here tonight, over my years at Yale. And it's to you guys that I really owe this very great honor. So I want to say thanks again to the Hope Funds for all that it's doing. It's wonderful. And thank you to all of you.